Good morning, everyone. The title of my presentation today is the effect of socio-demographic, lifestyle behavior, dietary habits, and living environment on the gut microbial diversity of indigenous Orang Ulu in Sarawak, Malaysia. I'm Associate Professor Dr. Priya Madhavan, and I'm from the School of Medicine at Taylor's University. So the human gastrointestinal tract hosts bacteria, fungi, viruses, and parasites. These microbes are collectively referred to as gut microbiota. These organisms have a symbiotic interaction with the host in which the host offers nutrient-rich environment, and in exchange, the microbiota provides benefits. So the factors affecting gut microbes are diet, lifestyle, environment, host-related factors, and also drugs. Therefore, the aim of this study is to study the differences in microbial diversity and its associated lifestyle factors among Kayan and Kenya, who are also known as Orang Ulu. These are the indigenous communities from Sarawak, which is a state in Borneo of Malaysia. So we look at the method. So this is the workflow of the study. The first part of work is the field work. The ethics approval was obtained by the National Medical Research Registry and Taylor's University, Malaysia. After getting the ethics approval, the demographic data and stool samples were collected from an indigenous community living in longhouses in Sungai Asap, Belaga Town, Bintulu, Sarawak. The stool samples were collected using OmniGene gut tubes. The second part of work is the lab work. So the second part begins with the extraction of DNA from the stool samples using QIAGEN stool DNA extraction kit, followed by purity analysis of the extracted DNA using nanodrop. After checking the purity of the extracted DNA, it was then stored at -80 degrees Celsius till further DNA sequencing. The third part of the work is the bioinformatics work. So this was based on the analysis of DNA sequencing. The DNA samples were sent to Apical Scientific, which is a sequencing company, for 16S rRNA gene sequencing to get the raw reads. The raw reads obtained from the sequencing company were completely analyzed, including the statistical analysis part in our labs using softwares, namely Quantitative Insights into Microbial Ecology known as QIME2 and RStudio platforms. The final part was based on the statistical analysis followed by report. So the target community were people known as the Orang Ulu. This is the indigenous community from Sungai Asap, Belaga, Sarawak. So this is a picture taken from the longhouses, and this is also the original picture captured during the sample collection for this project in 2019. So this is the demographic distribution of Kayan and Kenya ethnic groups in Sarawak, Malaysia. And if you look at the red boxes, so Kayan was the most number of people, and there were majority females, and also from the age group between 50 to 60 years old. So this was the household income. Majority of them had below 800 ringgit per month as the household income, and a majority of them were non-working. However, some of them were farmers and fishermen, and the rest were students or others. And this was their BMI, and their BMI was of normal distribution, majority of them, and the education level, majority of them were only educated till primary school. And this is the taxonomic. So this slide actually shows the relative abundance of the phylum and genus level. So for part A here is looking at the phylum level. So majority of it is from Actinobacteriodata, Bacteroidata, Cyanobacteria, Desulfobacteriodata, and so on. And in B, you can see the most abundant genus were from Eubacterium, Alloprevotella, Bacteroides, Escherichia, Shigella, and so on. So these are the most abundant that we got among these two groups of indigenous community here. So the next one is on alpha diversity. And in alpha diversity indices across dietary related factors. First of all, as per our analysis, the ethnicity, BMI, gender, occupation, household income, education did not reveal any significant association with alpha diversity. However, we do observe a significant association of dietary patterns with alpha diversity. So here in figure A and B, the number of meals, timings of meals, and bowls of rice exhibit significant association with alpha diversity. Diversity was significantly lower in those participants having more than 3 meals per day. For timing of meals in a day, the results reveal that participants who mention having meals in the morning, afternoon, and evening had significantly lower diversity and evenness of bacterial species. And bowls of rice were also found to be significantly associated with alpha diversity but have similar evenness. Participants who normally took less than 1 bowl of rice had significantly higher diversity than compared to those who took just 1 to 2 bowls of rice in their meals. The next one here shows livestock association or related to pets or livestock ownership against the alpha diversity. Factors including an appropriate number of years of animal ownership, cleaning of animals, place of animal defecation were significantly associated with alpha diversity as well. As per results shown in A, participants having animals for an approximately period of 2 years exhibited a lower diversity when compared to the rest of the groups of participants. However, we did not find any association with the evenness of species. For cleaning of animals, in B, alpha diversity was significantly lower in those who did not clean their animals as compared to those who cleaned their animals. Moreover, the place or animal defecation also showed significant differences in alpha diversity. People who mention that their animal feces indiscriminately have the highest alpha diversity among the rest of the groups. So in this diagram, here we can see that when we tested for health status on alpha diversity, we observed that alpha diversity was significantly higher in those who had received antibiotics during the last 3 months of sample collection. For the type of toilet facility they are using, as we can see in B over here, these participants who are using both types, pit latrine or pore flush latrine, have more diversity as compared to the others. So these are the notes or summary of what we just saw in the previous slides. So here in table 2, this is association again with alpha diversity with respect to the socio-demographic factors, lifestyle behavior, dietary habits, and living environment. So the table is a summary of all these values shown. So this table 2 shows a summary of all the values including significant and insignificant for alpha diversity indexes with respect to the socio-demographic factors. Food and water consumption, personal hygiene, pets and livestock. So for the analysis of beta diversity, we have employed Bray-Curtis dissimilarity index and weighted unifrac distances to investigate the structure of gut microbiota, and the principal coordinates analysis PCOA plots of beta diversity were generated using Bray-Curtis and weighted unifrac distances. So as per the results, there were significant differences in the gut bacterial diversity when considering participants who consume supplements as compared to those who did not consume supplements, and participants with probiotics and without probiotics. Participants having a number of meals as well as timings, and participants having different toilet facility. However, for all these factors, the amount of variability explained was low. However, you can also see in all these diversity images, there were no remarkable separations or cluster formation that was observed between the study groups as shown by the PCOA analysis. So this table shows a summary for all the values, including significant and non-significant values for beta diversity indexes with respect to socio-demographic factors, lifestyle behaviors, dietary habits, and also living environment. And then we come to the analysis of composition of microbiomes. So we have employed analysis of the composition of microbiomes known as NCOM to plot volcano plots to know which features were more differently abundant among the study groups. And from all the factors tested for NCOM, only 6 factors show statistically significant features between groups. For the factor of household income, only one differentially present feature from the genera Privotella was identified between the groups. Mitsuokela maltacida was differentially abundant in those participants who consume antibiotics as compared to those who did not. For supplement intake, Olsenella was differentially abundant in that group that consumed supplements during the past 3 months. However, Bilophila and Ruminococcus species, which were differentially abundant among those participants who had constipation problems. In this slide, NCOM results for bowel movement per day reveal uncultured Ruminococcus species that were differentially presented in those participants who mentioned no bowel movements daily. Moreover, the place for animal defecation also showed significant NCOM results. Uncultured Clostridium species were differentially abundant in those participants whose animals were defecating indiscriminately, that means in a random manner. So the study reflects the bacterial data dominated microbiota in these subjects. Ethnicity, gender, BMI, household income were not the determining factors for gut microbiota diversity and composition as well as observed in our population. Prevotella, Faecalibacterium, Succinovibrio were a dominant microbiota in the Orang Ulu subjects living in the longhouses. Prevotella in abundance indicates that Orang Ulu follows a vegetarian diet. Moreover, the larger proportion of Prevotella was most likely caused by the consumption of ulam. Ulam is also known as raw vegetables. It is eaten like a salad. Faecalibacterium and Succinovibrio abundance indicates a diet rich in plant-based. Depletion was reported in association with different inflammatory conditions. As per the analysis, animal ownership showed a significant influence on gut bacterial diversity. A significant association was also observed between approximate years of animal ownership and the gut bacterial diversity. An interesting observation is that these populations have almost no lactobacillus and bifidobacterium genera in the microbiota, even though they have this traditional culture to drink this fermented rice wine known as tuak. Escherichia shigella were also identified among the top 10 dominant taxa in the Orang Ulu population. And this is a matter of serious concern because these are associated with bloody diarrhea. The dominant representation of Prevotella, Faecalibacterium and Succinovibrio in the Kayan and Kenya gut is very appealing. It represents a potential source of healthy gut microbiota in fecal microbiota transplantation-based therapeutic strategies, especially in Malaysia, as they are involved in the production of short-chain fatty acids, the main metabolites produced by these microbiota in the large intestines. So this is the first of its kind to study and analyze the microbiota of the indigenous community of Sarawak and understand the link between the environmental and lifestyle factors and their gut microbiota. Rural communities also provide an excellent model to understand the influence of both natural environment and communal living on the microbiome. Yet, we hardly know anything about their microbiome. So the rapid urbanization in Sarawak is pointing out that it is high time to document the relationship between the biodiversity and the gut microbiome. Otherwise, these unique microbiome profiles will be lost. This is also because of the rapid cultural and socio-economic transformations that are happening in this state of Malaysia. These are my references, and I would like to thank you for your time in listening to my presentation.